So our next speaker is Dr. Mriganka Sur. Mriganka is the Paul E. and Lila Newton Professor of Neuroscience and Director of the Simon Center for the Social Brain at MIT, which he founded after 15 years as the head of the MIT Department of Brain and Cognitive Sciences. He has discovered fundamental principles by which networks of the cerebral cortex are wired during development and change dramatically during learning. His laboratory has identified gene networks underlying cortical plasticity and pioneered high-resolution imaging methods to study cells, synapses, and circuits of the intact brain. Recently, his group has demonstrated novel mechanisms underlying disorders of brain development and proposed innovative strategies for treating such disorders. Dr. Sur received the B.Tech degree in electrical engineering from the Indian Institute of Technology in Kanpur and his PhD in electrical engineering from Vanderbilt University in Nashville. Mriganka is also a scientific advisor to our board at the Autism Consortium. Mriganka?
Thank you, Peter. So it's a great pleasure for me to be here. It's wonderful to. It's a great pleasure for me to be here. I've been privileged to be part of the Autism Consortium right from its founding in 2006 now, and even I, as a hard-nosed, grubby scientist, when I look back, I think we have achieved a few things. My goal today is to share with you what we in the Simon Center at MIT are trying to do, and also share with you our vision. Why is it that we do, or that we're trying to do what we are doing? And we seek advice, and we seek help. So the mission of the Simon Center for the Social Brain at MIT is to understand the neural mechanisms underlying social cognition and behavior and to translate this knowledge into better diagnosis and treatment of autism spectrum disorders. Now, the idea that there is a social brain may seem unnatural or foreign to some of you, but in fact, the social brain is something that's very deep within us. Even a newborn infant, within minutes of being born, makes eye contact and sets in place. There is a built-in machinery in the baby's brain for social cognition. And this, of course, has not only developmental roots, but even evolutionary roots, as thoughtful observers like Michael Tomasello and others have pointed out. Children, babies, grown-ups are social beings. And so are there brain bases for social cognition, and hence for disorders that make social cognition go awry? Brain imaging has given us clues to the sensory and movement-related areas of the brain, which have been the first to be recognized. But then in the last 5, certainly in the last 10 years, there's been a revolution in understanding other parts of the human brain that deal with other higher-level stuff, including recognition of other people and complex objects in the world, and even ideas like personality. And importantly, one of the ideas that has emerged is that there are specific places in the brain dedicated to thinking about other people's minds. We cannot be social beings without having some image in our brain of what other people are thinking, or even when we have a conversation. We cannot have a conversation unless and until we understand not only what the other person is saying, but what's going on in that person's brain. Humans are unique in this respect in some way. We can think about other people's thoughts. And this happens progressively from children to adulthood. These are data from my colleague Rebecca Sachs showing how in very clever tasks called Sally Ann task, which I can tell you about, how when you make a person in a brain imaging scanner think about what somebody is thinking, children have difficulty, but then by the age of 5, they do it pretty well. And the brain regions involved in this task progressively then sharpen. So then this sort of helps us situate autism as very crucially a disorder of social interaction, along with other symptoms that the DSM-4, of course, everybody knows about, and the DSM-5 will change, as Walter has told you. But when we begin to understand the mechanisms by which the social brain is constructed or autism happens, we necessarily begin with a bottom-up view of mechanisms. And developmental brain disorders like autism have their basis in fundamental mechanisms by which the brain develops and is wired. Brain development starts in utero when the embryo is only 1 millimeter big, through waves of gene expression and molecular specificity that lays down the different areas that make the brain what it is, or that make the cortex what it is. These areas get wired up, and then inputs from the world as well as environmental influences help sharpen the wiring. Brain development is an intricate combination of genes and environments. There is no such thing as a gene that acts independent of an environment, but then every environment needs genes and molecules to be appropriately expressed. So it's reasonable to think about autism as having genetic roots. And indeed, we understand, and we have heard in this symposium itself, both rare and common variations in genes contribute to autism risk. We know a fair bit about the single gene variants, and you've just heard about neuroligin 4, but there are many other single genes, including the syndromic forms of autism that each have a single gene mutation underlying them. We have made animal models for studying these, and we have made some progress. To take things further, though, we need a deeper understanding. This deeper understanding, of course, arises from a better and better characterization of the human disorder through gene discovery, as you have just heard Steve Hyman describe, through models onto which the fundamental neurobiology of how the brain is wired and works has to be applied in order to understand why the disorder happens and how it gets expressed through specific targets that could possibly be targets for new or existing drugs individually or in combination that we can take back. The mission of the Simon Center is essentially out here. There is no disorder without or there is no pathology without a biology, and our mission is fundamentally to understand the biology and apply it to disease models. We do this by building the community of autism researchers, by providing seed grants to PIs and co-PIs. They don't have to be from MIT. They can be from anywhere, but we like them to be in the Boston area so that they can be part of our community. We give postdoctoral fellowships. We give research stipends to undergraduates. We want to catch them young. And we have multiple forums for building the community, including a very popular colloquium series. Some of our seed grants have been given to people across institutions. Bonnie Berger, a mathematician working with Zach Cohen, who is a bioinformatician at Children's Hospital. Gagan Joshi from Mass General, working with John Gabrielli at MIT. Sebastian Sung, who you'll hear soon, working with Guoping Feng. Bob Horwitz, a Nobel Prize winner, working on the worm, C. elegans, and Martha Constantine Patton, and so forth. More than 40 MIT faculty have some component of their research now dedicated to autism. But we cannot just solve autism by expanding the community of researchers, though that is a necessary condition, but it's not sufficient. What we need is to focus the mind on something that is potentially solvable. And for that, we take advantage of MIT's extraordinary strengths in developing tools and technologies through what we call hubs that are dedicated to building next-generation technologies that we'll make available widely to the community for mouse behavior, for generating human-induced pluripotent stem cell-derived neurons that you've just heard from Steve Hyman as well, and for taking brain imaging to younger and younger levels in the safer and safer way. And all of this we hope and expect to apply to two projects. We will not solve autism by saying we'll solve autism. We'll solve autism one mutation at a time. And the mutations that we have thought about, that we think will provide information, are these two, and I'll tell you why. But being at MIT, it's useful that we draw inspiration from radar. During the Second World War, a targeted project by the Department of Defense not only helped win the war, but helped transform the world as we know it. Out of radar came many, many technologies that shape our world today, including technologies for, say, encrypting data, or even for computers, or for signal processing, or many, many other things. So we regard these targeted projects as like mini radar projects. And that's just an MIT nerdy thing to say. So why SHANK3 and why 16p11.2, which you have also heard from Jerry Fishback? Some of the single gene mutations are beginning to yield. I've marked in yellow the mutations in which animal models have led to some mechanistic understanding of how the mutation in the Fragile X gene leads to dysfunction at synapses that leads to excess synaptic depression, which can be countered by a metabotropic glutamate receptor antagonist, as my colleague Mark Baer has discovered, and this is now in clinical trials, including in phase 3 clinical trials by Novartis. Rett syndrome is due to mutation in the gene MECP2, and my laboratory and others have shown that this mutation leads to excessively immature synapses, an idea first put forward by Walter Kaufman. And we have shown that by altering a signaling pathway between the gene and the synapse that passes through a set of molecules called PI3 kinase and AKT and S6, this pathway is downregulated, and by adding a molecule called IGF-1, we can restore levels of this pathway and thereby provide some efficacy to mice in which MECP2 is mutated. And this is now also in clinical trials, in a trial led by Walter at Children's Hospital. And of course, tuberous sclerosis, complex disorder, leads to a hyperactivation of a similar pathway called the mTOR-AKT pathway, and by using a cancer drug, we can downregulate this pathway, and this too is in clinical trials at Children's, led by Mustafa Sahin. But then, out of all of this comes the understanding that the synapse is a fundamental unit for understanding autism. Autism's developmental profile, the natural history of autism, is that it is expressed, at least, not right at birth, but perhaps a little bit later, where synapses are rapidly developing and forming and being shaped. And there are a lot of other reasons for thinking that autism may be at least one concept out of the 300 or 500 genes that might eventually be linked to autism, is that these genes must make molecules that meet at the synapse. And one of the quintessential molecules at the synapse is SHANK3. Many other molecules at the synapse have been already linked to autism, but SHANK3 provides a kind of a common pathway for thinking about how a synapse might be mutated, might be affected. And SHANK3 mutations underlie Phelan-McDermid syndrome, and a SHANK3 mutation, as all such synaptic mutations, are highly penetrant, which means that if a child has this mutation, that child will have the dysfunction that marks this syndrome or some other syndrome. So a very highly penetrant, highly expressed, specifically synaptic mutation is a useful thing to study as a way to provide a basis for integrating a lot of information. And this is a lot of words, but I just wanted to point out that our hope is to bring together 8 to 10 researchers at MIT, but also at Mass General and Children's, working on neural mechanisms, working on molecular pathways, and working on patient populations, as well as patient-derived neurons from their skin, which we can transform into neurons in order to understand what goes awry and why. And of course, we bring to bear technologies. Technologies are going to be transformative. This is a circuit that is hugely implicated in Phelan-McDermid syndrome, and that's because many of these neurons express the SHANK3 protein. And so my colleague Guoping Feng made a mouse in which he knocked out this protein, and he could show that there is less transmission from the cortex to the striatum. This is the part of the brain that's importantly involved in making movements and in making goal-directed movements as opposed to repetitive movements. And this pathway is built on the back of the SHANK3 protein that anchors receptors. When you knock out the SHANK3 protein, transmission is decreased in a significant way. And one example of how we might use this mouse, and remember, for the single gene mutations, mouse models are extremely useful because they do capture essential elements of the disorder. So, and as an example of technologies, something that I know well because my lab developed it, is to do extremely high-speed imaging such that we can visualize in identified neurons each spike that conveys information, and we can tell then between a normal mouse and a SHANK3 mouse what has gone wrong in the cerebral cortex when the mouse does something useful with its frontal cortex, such as a task involving executive function. Believe me, mice do have executive functions. You can train a mouse to, say, lick when it sees this set of bars and not lick when it sees this set of bars. But then when you cue it with, let's say, a green light, it will lick when this set of bars comes on, and of course not when this. But if you cue it with a red light, the mouse learns not to lick regardless of which bar comes on. So there is this executive control, and we believe that SHANK3 mice, or mice that have deficits in particular kinds of circuits, have deficits in such kind of control. And so in order to understand all of this, we need very high resolution imaging methods, and this is just an image of a set of neurons that combine two kinds of mice in which these are blinking, and essentially they are blinking because each blink is a spike, and this is in real time. You can see how spikes happen in a group of neurons whose identity you know as pyramidal or inhibitory or what have you. And this level of detail we think is going to be important for understanding exactly what happens in exactly what kind of circuits. We can of course alter activity by expressing light-activated channels and modulating the synaptic drive. So that's a little bit of glimpse into why SHANK3 and how we might understand the role of SHANK3 as a probe and as a kind of a critical point for many different mutations to feed into the synapse. But then the 16p11.2 mutation is a very different kind of mutation altogether. As you go from the rare single gene, highly penetrant mutations to the more complex mutations, 16p11.2, I believe, stands as a critical mutation in that pathway. And why is that? You've heard about some of this from Jerry, but this mutation was discovered both at Cold Spring Harbor as well as by the Autism Consortium in a wonderful study led by Mark Daly. So this gives me an opportunity to again embarrass Mark a little bit more. And it's a wonderful example of the collaboration and the power of collaboration across institutions that can yield the intellectual multiplication that, of course, that Mark does not need. Out of all of that has come the Simon's Variation in Phenotype project, and I copied down some of these numbers kind of shamelessly from a talk I heard by Wendy Chung a month and a half ago. And the main point I want to make is that this mutation is not nearly as penetrant as the SHANK3 mutation. Only 15% of people with this mutation show the full diagnosis of autism, though a lot of them are along the spectrum in some way. So you may have this entire region of 28 genes deleted, and you may show no symptom at all. And so this then begins to get us into thinking about what the other large subset of autism would be, because the large subset of autism is due to complex mutations that happen. Some of them happen in you and me. It's not as if you and me have this pristine genome in which we are completely normal, quote-unquote, people. But it's that some additional mutations or some critical mutations or some larger numbers of mutations add to tip us into the diagnosis of autism. 16p, I believe, is a critical step in that pathway because you need something else, usually in addition to 16p, to get the full diagnosis. What is different about these 15% of people? So So our questions then for the MIT group thinking about 16p, and here again there are about 8 or 10 people thinking about this. Why is the penetrance so variable? And are there additional genetic risk factors? What are the important genes within this region? What do these genes do? How do they contribute to all these different components of how the brain is built? And what is phenotypically, in terms of brain systems, in terms of brain imaging, in terms of behavior, unique about these cases with autism? So just a glimpse into how one might think about this. And this is picking up strongly on what Steve Hyman said. A mouse model may be useful, but will not be fully revealing when you have such subtle, multi-gene, complex, second-hit kind of disorders. We need models that engage human neurons. And so we make human neurons from skin cells or from other cells, such as liver or blood. So these are collectively called fibroblasts. But of course, making them from skin is the most useful. And you know that skin has a little bit of regenerative potential. Brain cells, once a human brain is born, once a brain cell dies, it's never reborn, except in two small regions of the brain. One is in the olfactory bulb in the sense of smell, and the other is in a place called the dentate gyrus that's engaged in memory. But skin cells regenerate all the time. You know that when you cut your skin, the wound heals. That teeny tiny bit of regenerative potential can be utilized to make neurons that have the same genetics in the dish. There are many, many different aspects of this technology that people are working on, including at MIT as well as at Harvard, how to go directly from here to neurons, how to make specific neuron types, how to make neurons that have the same genes except the one that is mutated, how to mark the two alleles. But very briefly, because I think Peter is getting up. Okay. I can go on this for a long time. So one critical issue in this field is how do you compare two sets of neurons? If I have autism and Peter Barrett does not, you cannot take my neurons made from my skin cells and compare it with Peter's neurons side by side, because there are many other differences between me and Peter. I'm vertically challenged. Peter is not. I have some hair, Peter has even less, and so forth. So all of this stuff may make our neurons differ. So that's not how you're going to solve this. You're going to solve this by taking neurons that do or that do not have, that are taken from patients who do or do not have autism, and induce a mutation in the ones that do not have, or read through the mutation in the ones that do and compare them side by side. So if I have a 16p mutation and I do have autism, you want to take my neurons and read through my mutation in the next dish and compare them side by side so that the rest of the genome is identical. It's mine. And so this is what Rudolf Jenisch is doing by either reading through or by inducing mutations. And that's why we call them isogenic neurons. And so here, even a subtle phenotype would give confidence. And then we can apply all of the stuff that you heard from Steve in order to do screening for compounds to see how can you fix it. But there are a couple of other things in 30 seconds or less. One of the major issues in this field is how do you make different cell types? The brain has at least 500 different cell types. And so Fatih Yannick, a very talented electrical engineer, can blast through, is making tools for blasting through billions of combinations of transcription factors in the dish to see how can you make inhibitory neurons versus excitatory versus somatostatin versus parvalbumin versus neuropeptide Y subsets of neurons. And you want to capture as closely as possible the brain in a dish. So we can make substrates in which we pattern proteins layer upon layer upon layer upon layer to make a three-dimensional scaffold in which human neurons can be seeded so that they help wire up in certain ways. And through all of this then, one can make circuits in a dish that we can probe with probes of synaptic structure and function. And they're actually human neurons wired up into circuits. So in conclusion, autism needs to be solved one mutation at a time, but hopefully This will yield broader concepts. We, of course, need to understand what are the genes, and whole genome sequencing and large patient samples are critical. We need to understand what do the genes do, not only by making models, but also putting human neurons in a dish and even human neurons back in mice, because we want to have as clean an environment as possible. What I haven't told you, but what I started out with, is the social brain. One of the great challenges and gaps in the field is how do these genes map onto the brain systems that make the social brain? And that is a very significant challenge that will involve not only mice, but then perhaps human brain atlases and even monkeys as an intermediate step in terms of trying to understand why social phenotypes in subsets of autism. I'm kind of a skeptic about biomarkers, but that's a different story, because I believe that very complex disorders will not have very ubiquitous biomarkers, which leads me to the concluding point that ultimately we will solve autism one patient at a time, but based on the concepts that we derive from model systems. Thank you very much.